block. We just want to have a good understanding of basic microbial genetics, how they replicate their DNA genomes, how they express genes, how they acquire or change their genomes, genetic variation. And then we'll go over some basic terms to get started. We spent a little bit of time talking about structure and function of nucleic acids, DNA and RNA, which I think we can kind of accelerate through because you are doing this simultaneously in biochemistry as well, right? Okay, cool. So fast track through that a little bit. I'll review. Sweet. Okay, we'll take it. We'll see. So we'll go through that. Once we know what these things are, then we'll kind of talk through these processes. We'll talk to replication, talk to transcription, translation. Then talk about gene expression, again, in a prokaryotic setting. And then talk about genetic transfer mechanisms, a little bit about plasmids, and some of these specific mechanisms that allow bacteria to, again, change their genetic makeup a little bit. Okay, so getting started here, very basically, this schematic is showing you what we call the central dogma of molecular biology. Sometimes it's called the central dogma of life. All organisms, prokaryotes and eukaryotes alike, will adhere to this basic schema here, where when they need to replicate DNA, they're doing so in preparation for cell division. So they'll replicate their DNA. And when it's time to express their genes, then they have to undergo transcription. So genes are transcribed into RNA, messenger RNA, and then those messenger RNA transcripts are then in turn translated into protein. That's how it works. So basic core concept here, just initially, you don't want to conflate what's going on with DNA replication, which is again just making copies of the genome in preparation for cell division, with gene expression, which is the rest of this stuff. And again, common to all life, everything needs to be able to transcribe genes and produce proteins. In terms of nucleic acid structure, this schematic diagram on the left here is kind of just showing us double-stranded helix without the helix, just laid out flat or planar. And on the inside of this helix, you can see we have our actual bases, 4 different bases, A's, T's, C's, and G's. You can see that they're bound together by hydrogen binding in the center there, double bonds between the A's and T's, and triple bonds between the G's and C's. So that's in the center, bonded base pairs. And then moving outward from that central part of the ladder, we have this sugar phosphate backbone. So the sugar is ribose. It's a 5-membered ribose sugar molecule. And this carbon on the sugar molecule is what is bound to that base. And this carbon on the sugar molecule is what is bound to a phosphate group, or another carbon that's coming off of this 5-membered ring. But this phosphodiester backbone is what is actually linking all of this together on that backbone. In terms of how our ribose sugars actually look for DNA, this is how they're numbered conventionally. 5-membered ring, but there's an oxygen instead of carbon in this position. And then we start counting a number in our carbons going clockwise from there. So that first carbon on that ring is the C prime carbon, carbon 1. And that is the carbon where that nitrogenous base is coming off of in the center there. Then we have our 2 prime carbon, which is really important because that determines whether it's going to be DNA or RNA. Talking about DNA, that 2' carbon will just have a hydrogen coming off of it, a proton, essentially. 3' carbon here is important because that has a hydroxyl group coming off of it. And that's regardless if it's DNA or RNA, the hydroxyl group links, it's part of that phosphodiester backbone. So it's important for that. 4' carbon is a 4' carbon. And then the 5' carbon of note, this is not actually on the ring, it's coming off the 4' carbon. So it's connecting the rest of this ribose molecule to the phosphate group. So, this is just another schematic. On exams, you could be asked to identify a number of different what's going on with each of these carbon groups, 2' carbon, 1' carbon, things like that. So if I asked you, like, what is special about the 1' carbon, not that I would necessarily ask you that, what is that doing? It's binding to the nitrogenous base. Yeah, exactly. Okay, so looking now, we see that helical turn is a little bit more realistic about, yes, sir. Exactly. So if it's RNA, and we'll have a full RNA slide, but DNA, what does the D stand for in DNA? Deoxy. Yeah, so there's no hydroxyl group in DNA, it's a deoxyribose. RNA, it's just ribose, so it has that hydroxyl group, not that 2'. Yeah, but that is, just by looking at the ribose alone, that's how you can tell the difference between DNA and RNA, that 2' carbon. Very important that you remember that. Okay, so we've got our double-stranded helix. You can see it is twisting, very well characterized molecule. You can see those bonds, those hydrogen bonds between those different bases again. You said how many hydrogen bonds between A's and T's again? 2, and then for C's and G's? 3, yep. I corrected this on the slide. It should be attached to the deoxyribose sugar phosphate backbone, a little bit more specific detail. So just add the word add the phosphate in there if you will to your slides. In terms of what the A's and T's and G's and C's stand for, we have adenines and thymines and guanines and cytosines in the DNA. The A's and G's, adenines and guanines, are purines, meaning that they are double ringed structures, and the T's and C's, thymines and cytosines, are pyrimidines, so they have a single ring structure. Fortunately for this course, you don't have to remember that, but it is important to know that a purine is always going to bind to a pyrimidine, right? So that's why you have A's and T's and G's and C's. It's the way it always is with DNA. In terms of orientation, there is an end to each one of these strands. There's an orientation. So if you were to have looking at this as a linear molecule that suddenly were to be discontinued, that last carbon on that phosphodiester backbone, we would be either a 5' or a 3', and that refers back to this structure, either 3' hanging off that, or the phosphate, the 5'. But that's what we mean when we say orientation. And that's only if it's a linear structure. If it's a circular DNA molecule, there's no end. If you cut it, if you have like an endonuclease cutting one of those strands, then you can have a 3' or 5' end. Okay, for a replication, very simply, as a basic overview, when you start off replicating a double-stranded helical molecule like that, first things first, you've got to separate out those strands, right, which are very stably bonded together with those hydrogen bonds. So that doesn't happen on its own. It's going to take some enzymatic assist for that to happen. You can also force this to happen by applying heat, which will cause those hydrogen bonds to dissociate from each other, or you can do it chemically with high pH, low pH, or you can salt those bonds to separate. But in the cell, when the cell is getting ready to actually divide and it needs to do DNA replication, this is all enzymatically handled by the different macromolecules and machinery. So the first one that we're going to talk about is helicase. This particular enzyme is responsible for providing that catalytic energy to dissociate the hydrogen bonds between those bases and allow them to separate. When you have a circular DNA molecule, like a bacterial chromosome, right? When you start opening that up, what that does is it increases the tension throughout the entire molecule. It's called torsional stress. So imagine like a phone cord, for those of you who remember what phone cords are. If you twist it, it starts getting really, really tight, right? And that's bad because eventually you can twist it so much that it starts to break. So in order to alleviate that, you have a molecule called gyrase, which is a type of molecule, like belongs to a big family called topoisomerases. And a gyrase will come and do a double-stranded nick elsewhere in that molecule to make sure that the torsional stress doesn't cause breakage in the DNA strands as it's being replicated. So that's what gyrase does. And then we have a replication fork forming as this helix unwinds, so we can actually expose this DNA that needs to be replicated. In bacteria, we have our circular chromosome up here. You can see that cartoon here. This is called the origin of replication. On that chromosome, there's usually just one ORI, origin of replication. And once you get this fork initiated, this is what's showing in that second panel there, it goes in both directions. So it's very efficient, but you'll have replication proceeding to the left, so to speak, and replication going on to the right. And that'll happen until that entire chromosome is copied. Sometimes when that copying is done, those two new circular chromosomes are linked together. That's called concatenation. And then they have to be enzymatically cleaved to separate. And sometimes they're automatically dissociated, but either way, the end product is you have two completely new chromosomes. So old strand, new strand, old strand, new strand, that can then go on to the daughter cells. So when this bacterial cell is replicating, it'll split into two, and then one copy will go here, and one copy will go into the other cell. In terms of the enzyme that actually does the replication part, the workhorse of this is called DNA polymerase III. So it does the vast majority of actually adding new nucleotides onto that growing new strand and facilitating strand growth. This cartoon it's simple, but I think it's one of the best ones to kind of walk through this process. So you have your replication fork forming, right? And this DNA polymerase III needs to start adding these new nucleotides on to build up this secondary daughter strand. However, polymerase III can't just come and add new nucleotides. It doesn't work that way. It can only add them to the 3' end of that strand. So to overcome that problem, you have another specialized type of polymerase called a primase that comes along. And the primase with prokaryotic cells will lay down a little bit of track, if you will, like a little bit of railroad track. It's usually like only 10 to 15 nucleotides long, but it's an RNA primer, and that's what's kind of depicted. It's what's depicted in green here. So this is an RNA primer laid down by primase, and that will leave a 3' hydroxyl group exposed for that DNA polymerase III to build off of. So it'll build all those new incoming nucleotides off of that end. Because of that, once it gets going, it just continues all the way down towards this replication fork, smoothly proceeding along. This is called processivity. It goes at really great speeds because it has a lot of genes to get through. I'm sorry, not genes. It has a lot of bases to copy during this process. But this is happening on the leading strand, going towards the replication fork. On the other strand, it's a little bit more complicated. Because replication can only occur in the 5' to 3' direction, adding to those exposed 3' hydroxyl groups. On this strand, it's going to be significantly more complicated. You still have the major players like primase coming down to lay down this 10 to 15 nucleotide primer, and you still have DNA polymerase III doing the yeoman's work of synthesizing that new strand. But because you have this problem of laying down only 5' to 3', you have to do this in a discontinuous, disjointed manner, if that makes sense. So you're kind of moving away from the replication fork, but you're still going in that 5' to 3' direction, and you still need all these different RNA primers to be laid down for that polymerase to synthesize from. These fragments of new strands are called Okazaki fragments. This cartoon only shows them as being like 7 or 8 nucleotides long, but in reality, for prokaryotes, they're usually more like 1,000 nucleotides long. And that polymerase will do what it always does. It just keeps adding nucleotides until it bumps into the next Okazaki fragment down the road. And then it basically just falls off through steric hindrance. It bumps into that fragment and it falls off. So then you have a problem. On this strand, you now have a couple problems actually. You have a gap. You have RNA still hanging out there that needs to be removed. And then it's not connected, so it's not a continuous piece of DNA. So all that has to be fixed. So how do we fix that? We have our synthesis of those Okazaki fragments. Then we have another type of polymerase that has to come in called DNA polymerase 1. And what that does is two things. First thing it does is kick off the RNA primers. It gets rid of those, so you remove the RNA primers. And then it has to fill in the gaps with actual DNA nucleotides, and that, however big that gap is, needs to be filled in by DNA polymerase 1. So problem solved, right? No. What else has to be done in order for this to be?
Good, join them.
You got to connect them together. So then your last important player is this ligase. What does it mean to ligate things together? Connect things together, exactly. So ligase is another enzyme that will join those fragments together, and the resulting product is you finally have that piece of continuous new DNA. Does that make sense so far? Okay. All right. Then we have this very complicated picture. If that last slide made perfect sense, then hopefully this will kind of also make sense. Okay, so what's going on here, we have a GIF that's kind of showing this happening, that we have two replication forks going in different directions from the origin of replication. That's what the GIF is showing. And then here in the upper right inset there, and we have a nice holistic view of that bidirectional replication fork. And then we're zooming in on the one on the left. That's, for whatever reason, more intuitive to understand. So zooming in on that portion, that left replication fork, what we're not seeing here is that there would still need to be an RNA primer, but that polymerase is basically synthesized continuously on the leading strand, at that new strand. The only other players that we haven't talked about are the sliding clamp here, which actually forms a complex with DNA polymerase 3, and that basically just keeps the molecule stable as that DNA is being fed through it. Otherwise, the RNA polymerase would probably just fall off and lose its plasticity. So you have a sliding clamp that's needed. Helicase, we already talked about, is doing what again? It's facilitating the dissociation of those hydrogen bonds. It's unwinding the DNA. We haven't talked about single-stranded binding proteins. Those will help keep the single-stranded DNA separate from re-annealing until that process is done. So it'll also help protect it from any nucleases because single-stranded DNA is very vulnerable to nucleases. So it has a protective function. What else? What else have we talked about? There's our gyrase. The gyrase is doing what? Yeah. It's releasing tension by making some mix.
It's not really talked about, but the the leading strand, once it's complete with it has the RNA polymerase primer, or the RNA primer on it that started the DNA polymerase, right? So that has to be removed from the leading strand as well. Like once it comes all the way around.
Oh, okay, yeah. Yeah, so on the leading strand here, like up here on the top of the fork, right? That will get removed, and it will get removed by that same type of polymerase, but it's DNA polymerase 1 that has the exonuclease activity. Yeah, but it's treated like an Okazaki fragment in terms of removing that RNA primer and sealing that. Yeah. Okay, so yeah, leading strand, easy peasy. It's moving 5' to 3' towards the replication fork. Lagging strand, a little bit more complicated. Got to do this piecemeal. But the reason why I like this figure is because it shows, as we move to the right, how things kind of get perfected. Right? They get sealed together. And you go back in time, you see DNA polymerase 1 doing its thing, which is kicking off the RNA primer, and then adding DNA nucleotides in there. So it's kind of nice to just pull all this together. Any questions?
Then what did you say the sliding clamp did?
So the sliding clamp, it remains in complex. It's complexed to the DNA polymerase 3, and it keeps it on track. If it weren't in association with that sliding clamp, which, like when DNA replication occurs, the sliding clamp will open up like a hand, and then it will close right around the DNA molecule so that DNA polymerase can stay associated that entire time. If you mutate it or make it so its active site doesn't, bind to DNA, that polymerase just keeps falling off if you don't get anywhere. So it's necessary for processability.
I have the same question.
Okay, yeah. Does that make sense?
It does.
Okay. All right, any other questions about this figure DNA replication? Okay. All right, so what we have here is kind of like a nice representation of what ends up being the final result of this type of replication. It's called semi-conservative replication. It took a lot of years to figure out that this is the way the DNA is replicated, and I think there was a Nobel Prize awarded for it in 1958. But essentially, when you have your two old strands, the daughter cells will get an old strand and a new strand, right? And then this continues on. Let's see. There is editing that is done during replication, and it's usually pretty high fidelity editing as well. When I say high fidelity, what does that mean?
Pre-edited.
Exactly. Yeah. Like a high fidelity recording of something means that it's like a good recording. So it has to be, because if you make mistakes during cell division for one bacterial cell, what happens?
Dies.
It could mutate.
Yeah, they're usually going to be what we would call deleterious mutations. So if you make a mistake, it needs to be fixed. And we have polymerases that have that kind of proofreading activity and that can fix those mistakes as they occur. And those, like I said, they're higher fidelity than other, than like transcription polymerases. Okay, so that was just like a 10,000-foot view of what goes on for DNA replication in cell division. Let's completely like shift over now to talking about what happens when you need to express genes for bacterial cells, for prokaryotes. So here you have our genome. We have a gene that needs to be expressed. So we transcribe it into mRNA, and then again that gets translated into protein. Just a note here on the slide. The polymerase that does the transcription part is going to be named an RNA polymerase because it's making RNA, but it's DNA dependent because the template is DNA. Make sense? DNA dependent RNA polymerase. Before we talk about transcription in any kind of detail, I think now is probably a good time to talk about RNA and how it's different again from DNA, which we already sort of talked about a little bit. So, for example, we already talked about the fact that this 2' carbon on the ribose is really important in distinguishing DNA from RNA. But in terms of the bases themselves, when we have DNA, we have ATCG, but when we have RNA, what are our 4 bases?
AUCG.
AUCG. So we have a uracil instead of thymine. And in terms of the differences here, there's just a methyl group coming off this carbon with thymine, where this uracil is just a protonated carbon. So it's a very minor difference, but it makes a lot of makes a huge impact on the way this molecule behaves. With the DNA double-stranded helix, we can see again very stable molecule, energetically stable. When you have a single-stranded RNA molecule, this is a very unstable molecule. It does not do well. So most of these RNA molecules that we're going to be talking about, mRNAs, tRNAs, and RNAs, they're meant to be pretty temporary. They don't last for a long time. DNA can last, if you preserve it well, it can last for hundreds of years. You know, it can last for a very long time. But RNA has a much shorter half-life. The other thing to discuss with RNA is the fact that since it's not annealed neatly into this, hydrophobic core like DNA is, those bases in the center rungs can stick to everything. It's like a piece of tape that you're wiggling around, single-sided tape. So when you do that, it will anneal to itself, and it will become difficult to control, okay? Because those bases base pair to each other. Extensive intermolecular base pairing. All right, and then this is just a completely redundant slide, but let's see. Here we're showing a DNA molecule with a proton coming off 2' carbon, and again, in this cartoon, it's showing you an RNA nucleotide with a hydroxyl coming off 2' carbon. Any question about that? No. Okay, so that's like the quick and dirty on RNA structure. Getting into messenger RNA now, so we can talk about how transcription occurs. When we have that mRNA transcript that's coming off of the DNA genome, it is coded in such a way that it can be read specifically by ribosomes and translated. This code is called the codon code, which is a triplet code. But when this transcript is read in a linear way like this, and each of those bases are read, they're read in sets of 3. So each frame of this code is read in sets of 3, and if you shift that by either 1 or 2, you completely mess up the message. It has to stay in frame like that. So GCU, ACG, GAG, if you were to add or delete some of these nucleotides in multiples of 1 or 2, again, that would completely screw up this nice little code that gets read by the ribosome. Each one of these codons has a corresponding amino acid, with the exception of start codons and stop codons. Those just tell the ribosome whether that gene is, whether to start translating or whether to finish. Okay, so this is what I was mentioning with the codons being arranged into a specific reading frame, and that there's only one reading frame that is sensical for protein translation. And if you shift that, then you get a non-sensical protein that's translated. We only have 20 amino acids, which are in this chart, which you guys have seen before, right? The codon chart, everyone's seen before? Okay, good, so you know how that works. We only need 20 amino acids, but if you take all the possible codons by multiplying each position by 4, 4 times 4 times 4, you could generate a total of 64 combinations, 64 codons. If you look at the codon chart, you only have 20 amino acids in there, which means you have a lot of redundancy. And that redundancy has a word, specifically it's called degeneracy. That just means that you have multiple codons can still code for the same amino acid. So we'll talk about that in a little bit more detail here. All right, so a little check on learning, make sure we're tracking. We have some examples of how to read this codon chart, see how you guys do. All right, so if you have a UUU, the answer's up here for you. You have a phenylalanine, but how are you reading this chart to get that amino acid?
Left side, top, right side.
Exactly. So it tells you first base and codon. So the first base is a U, the second base is a U, so up here it's labeled second base. So we know we're going to be in this quadrant right here. The third base on the right axis there is a little trickier because its pointing square is sharing all 4 nucleotides, but the top one is a U. So if you follow that across, there's going to be a phenylalanine. UUU corresponds to phenylalanine. All right, what if we shifted on that first base and went from a UUU to a CUU? What would be the resulting amino acid?
Leucine.
Leucine, exactly. And then AUU,
Isoleucine.
isoleucine, and GUU,
Valine.
valine. Okay, cool. So basically, we just shifted all the way down on this left-hand side of that codon chart. If we move to this part of this practice thing, we're just mutating that second, or we're changing that second position. So we're doing the same thing. We're just moving along, staying in this first row for U. We're moving from UUU to UCU to UAU to UGU. Okay. And then, so real quick, can you rattle off those amino acids for the second row, second position?
Serine, tyrosine, cysteine.
Good. Yeah, phenylalanine, serine, tyrosine, and cysteine. And then lastly, I know this is, we're sort of beating a dead horse, but when it comes time for exams, it's a good thing. Okay, and then if you're just moving the third position, what are those 4 amino acids if you're just changing the third position?
Both leucine. Phenylalanine the first 2 and leucine the last 2.
UUU is
Phenylalanine.
phenylalanine, UUC is
Phenylalanine.
same, yeah, because we're just now staying in this upper left square, right? We're not going anywhere. And then UUA,
Leucine.
leucine, and leucine. Okay. So in sum, basically, if you change that first or second position, you will probably get a resulting change in amino acid. That third position is sometimes called the wobble position, which means it can tolerate changes better. You have a 50% chance of mutating that third position and still getting phenylalanine, and you have a 50% chance of mutating a leucine and still getting leucine in that third position. And that is to protect against mutations that happen. Most of these that are grouped together on the codon chart have similar characteristics. For example, phenylalanine and leucine in the upper left, those have similar characteristics. If you have a mutation and you switch from phenylalanine to leucine, there might be survivable changes that occur. Okay, so going back to transcription, zooming back out now that we know how the codon chart works, the codon system, we have our DNA. It's getting transcribed into messenger RNA transcripts, and then it's getting translated into polypeptides, proteins. But on that DNA, now we actually have a difference between those strands, and they're important to know what those differences are. So these are complementary strands. The top one here is called the coding strand, and the bottom one in blue is the template strand, also known as the antisense strand. When you have a transcript coming off, it's going to be reading from the template strand, as the name suggests, but make sure that is straight in your head, that it's the template strand that the RNA polymerase is going to be reading from. So here's the RNA polymerase. It's reading that template strand in blue. You can see this step here. So when there's a T, it's going to find an A and make sure that it's bound that way, and so on and so forth, all the way down into that transcript is released. Okay, so a little bit of a check on learning for you guys. If you have a sequence on a coding strand, which is, what color is it, orange or blue here? Orange. Okay, so if you have a sequence on the orange coding strand that is CGT, what would be the corresponding sequence on the transcript?
GCA.
It would CGU. Yep, exactly. So U because it's RNA again. And if your sequence on the template strand in blue is TAC, what would be the corresponding sequence on the transcript?
AUG.
AUG.
Start.
AUG, exactly. So now it's complementary. All right, transfer RNAs. Talk about those a little bit. So the transfer RNAs are the molecules. These are RNA molecules that are actually going to be responsible for matching the sequence on the mRNA to the appropriate and correct amino acid. So in order to do that, they need to recognize on this portion, which is called the anticodon stem, they need to recognize that specific codon by a complementary sequence called the anticodon. So the anticodon is sitting on the bottom of that anticodon stem, and at the top of that tRNA molecule, top in quotes, the top you have the actual amino acid itself. So in this case, if you have an anticodon, let's say its sequence is UCA, that is what you would use for your codon chart to actually determine what that amino acid is that it's corresponding to. Okay, so if you look up AGU, the codon on the mRNA in that codon chart, you're going to get the wrong answer. It has to be the anticodon. Okay. So what is the, which one of these, AUG or UAC on the second one, is corresponding to tyrosine? Codon or anticodon?
Codon.
Anticodon. Yeah. AUG. You can confirm that and look it up here on the codon chart.
AUG is methionine though.
AUG?
That's a star codon, it's methionine.
Oh, you're right. Okay, I'm sorry. I think there's an error on that. I will correct that. Yeah, sorry about that.
So there's no, so we follow the anticodon. I was seeing on the 5' side yesterday with serine, but so the 3', it's still anticodon, so it doesn't change, like, because that's AUG, you would follow the codon. That's irrelevant, like it's always anticodon.
It's always anticodon, and that's because of the complementarity. Yeah, exactly. Yeah, so those tRNA molecules, I mean, again, like all their purpose is to do A, matchmaking, make sure like this is the right amino acid to add, and then B, actually bring in the amino acid itself. Like this is physically the serine that is coming in to be added to that chain. Next. Okay, last flavor of RNA that we're talking about is the ribosomal RNA, the rRNA. And this is going to be the portion of the ribosome itself that does a lot of the recognition, the site where the codons and anticodons are meeting. With prokaryotes, these ribosomes are made up of majority ribosomal RNA, about 60%, about 40% protein. So these rotating structures here will show you these two big subunits that comprise a prokaryotic ribosome. But the one that sits on top is called the 50S ribosomal subunit, the large ribosomal subunit. And the one on the bottom there that's responsible for decoding that codon message is the 30S subunit. When you're referring to that ribosome together, the 50S and the 30S, it is called the 70S, which doesn't add up, does it? That's okay, because it's not a unit of mass, so it's not meant to be additive. With eukaryotes, with us, we have 60 and 40, respectively, for large and small ribosomal subunits, and those add up to 80, right? So again, if you're thinking they should add up because they're units of mass, they're not. They are Svedberg values, which is like an indication of how rapidly they settle to the bottom when you're centrifuging them. It's a sedimentation rate. Okay. So again, 40 and 60 with eukaryotes will be still referred to as an 80S ribosome, and for prokaryotes we call this 70S ribosome, even though it's 30 and 50, small and large, respectively. And this is just an FYI if you're ever wondering why a 70S is called a 70S. Okay, so in terms of transcription, we'll kind of break this down again, very minimal detail, but just so we have an understanding of how transcription works. Actually, I've got one for you. Why don't we take a 10-minute break and come back, and then we'll talk through the transcription.
That's me. I probably should get a suit and text. Huh?
No. The tRNA is already bonded. It's just like, imagine it's just like out here, floating around. It's a loose form. It's just out here without all different types of amino acids. This tyrosine is serine, isoleucine. Like they're all just floating around. All the tRNA is already bound together. It's like the one that because it has amino acid, it's going to express this anticodon to bind it. And like that activation. Right. Because the tRNA is already floating around like this on it's going to have reciprocal binding. It'll match up.
I feel like it's a big deal.
crazy about this.
Well, she said something that I don't, I'm gonna ask a question. Once the group chills out. It's like, something about fine, based off any code on it. That's not how it's I'm always using based off of RNA. And so like AG would be series, right? And then series. And then
You're running low on your mustard, please, Steve.
but then we should reusing anti-codon, right? Because then you can get used to it. You still get series. That's so like we're like maybe get combined, then you can use people are like, oh, that's science.
What do you know? I don't know. Maybe when you're in
It's all your fault.
How I got
I thought you were talking about
codon. That's why I'm like, wait, what? I'm gonna ask Chad.
deep that. I'm sure that's what these guys are doing.
Maybe. Yeah, maybe.
So either we're going to talk about biochemistry or
Because if you translate it, if you take the transcriptions, and you make the transcriptions, and then it gets translated. So the transcription is what's coding for the anti-coding just connects it. But the codon
Dude, I can't believe we're mouse confused.
so this in terms
But that would be.
Oh, really?
Because why would that tyranny be around telling them to stop? Transcription.
I thought it was again.
Yeah, that's right. Like you're like RNA. You use one transcribe. You use that, but if an mRNA gets have an mRNA, that means to start you're never gonna start. There's gonna be a peak with like let's say like the anti-coding or let's say let's say
You guys, real quick, I know you're on break, and I'll say this again after break, but I know a lot of you were talking about this, or I was wrong about this. No, I was sorry, I was wrong about the codon here. This figure was a little confusing. It's not the anticodon, it is the codon that is supposed to be corresponding. And I think what tripped me up is, unfortunately, UCA
it does
corresponds
also
to a
correspond to the
serine.
same thing.
Yeah, where AGU does as well, so it's kind of a horrible example. But on this one, I thought this was incorrect, but that tyrosine is correct because the codon, the UAC, does code for tyrosine.
You see that, right? You see that.
It's just a codon, yeah. And it kind of, yeah. So I apologize for that. I will say the same thing again.
Yeah, imagine tRNA just floating out.
So you read the bottom. So it's crazy because serine, UCA, and AGU both code for serine on this, right?
So imagine
So it's UAC codes for tyrosine, UAC codes for tyrosine, but UCA, serine, and also AGU is also serine. So this one you could read either the anticodon or the codon, but how it reads it is the codon is read by the tRNA or by the ribosome, right? It pulls in a tRNA.
part of that ribosome. I'm sure we'll go over it. Probably I'm going to show you. So yeah, you see this. So this is like it's getting the polypeptides that's getting created right now, the protein. This one is these TNA that got brought in. Some enzymatic action is going to happen, and it's going to be cleaved off of this. This whole polypeptide is going to be cleaved off of this one. That TNA is going to figure out what's going to add to this one. And then a new TNA.
Fuck yeah.
Yeah, one second exit site, just like the last one. Yeah. Yeah, like it comes in the middle one.
She said that the protein
Like nothing about it binding to RNA that brings amino acid onto the RNA. It's already bonded to RNA. When it comes to the ribosome, what's happening is that's not getting added.
This one already.
Remember that video that I showed you? Where the ribosome looks like this. It has like a large subunit and small subunit of mRNAs here. It has the 3 little binding sites where the tRNA comes into. Remember how I showed you there's the insertion, the exit, and then the U site? And this is like, it reads these 3. So it comes in here, this tRNA comes in here, it reads these 3 codons. And then the ribosome shifts over, so now these 3 shift over to here. And that's where the tRNA, you have your peptide, right? Another one comes in with the peptide, that makes the peptide bond, and then this tRNA leaves. Remember how I showed you that video? So that, it reads the codon right here on the end line. So it reads this, not this. She said it shouldn't be a problem for me to take it tomorrow. Huh? Said it shouldn't be a problem for me to take it tomorrow. Oh, it's gonna be a long
Right
day.
now, it's obviously like it's folding as it's coming out, but because it's still attached, there's still a chance that it's maybe all the way +2. And then maybe it's inactive when it gets released too. And it would require another enzyme to pull it in, change it to an acid. You know, much like something like you know, like in the stomach, it requires a certain enzyme or a certain pH to be active, right? So like, not just because it's going to get attached to an active enzyme or a protein. Whatever. All I'm saying is that like when it gets attached, it's a polypeptide. It's like a protein. It can be a bunch of different things. Depending on like what's the or it could be folded.
Hate to see it.
Clean up my hair a little bit. I didn't get to blend it. I had to clean it up because it was getting. Dude, I was bad before she did that haircut. I'm gonna let it go for like 3 weeks. I didn't touch the top. Just the side.
Oh yeah. Yeah, I did
Right here. It's like just that. How we talking about? Yeah. You know, Memorize this chart. I know each one.
Okay, guys, let's get going again. All right. Okay. This dreadful slide, yes, I said this during break. It is correct, all right? And it is the codon, not the anticodon, that determines the amino acid that's brought in. I'm so sorry about that. It's just unfortunate because, like I was saying during break, UCA also happens to encode for serine on that stupid codon chart, but it's the codon. As the name implies, codon chart, codon, easy to remember, we will never forget, right? This entire room of people, myself included, codon chart. Okay? Okay. All right, so let's drive on then. Talk about transcription. All right. So now that we know what RNAs are, and tRNAs, and messenger RNAs, mRNAs, let's talk about how this all works together in prokaryotes to actually express genes. All right. So we've got 3 different steps. We've got the kickoff, the initiation. We've got the elongation of that mRNA transcript from the gene itself on the DNA. And then we have to have an end to this. We have to have signals that tell that ribosome that that's the end of the gene and you can terminate translation. Okay, so initially what happens is you have this RNA polymerase and it will need to bind somewhere upstream of that gene that is going to get transcribed. So there's a promoter region that it will bind to. The gene itself would probably be somewhere in between that promoter and the stop sequence that it will read. That's in green there. And then similar to when we have replication, you need to have access to the gene itself. So there's an unwinding that occurs, and that unwinding is also facilitated by different molecules that will make sure that those hydrogen bonds are loosened. And then immediately after transcription, they just kind of re-anneal back together again into a double-stranded molecule. So unwinding, there's a transcription bubble that forms, and that just allows enough space for that polymerase to do its thing. There are a bunch of ribonucleotrophosphates that are around that can be added to that growing chain. So during elongation here, you're moving along, synthesizing that same direction, 5' to 3', adding new nucleotides along. And when that RNA polymerase, this big purple blob, this molecule enzyme, hits that termination signal, it will terminate. But up to that point, this transcript keeps growing. This single-stranded RNA, messenger RNA transcript will grow. Because these genes are pretty big, they're around 1,000, maybe 2,000 bases or so, this has to occur pretty rapidly. So these new nucleotides are added on at a rate of around 40 per second. 40 to 90. And this is much, much faster than eukaryotes. We have more complicated architecture in the way our genes are packaged in chromosomes. So with us, it's probably twice as slow, but we still process with RNA polymerases moving as quickly as possible. Okay, so same direction as before, transcription occurring 5' to 3' and maintaining high rates of processivity. Also, I mentioned when we were replicating DNA that you have some really good, robust proofreading mechanisms involved with DNA replication. When you're talking about RNA transcription, those proofreading mechanisms are not as good. So you have a higher mutation rate during transcription than you do with DNA replication. And that's because of the RNA polymerase that has less robust proofreading mechanisms. All right, when it does hit those stop signals, it'll codon that tells it you can stop, and that RNA polymerase will sit there and it'll pause for a second. When it pauses, it's kind of like riding a bike and you lose momentum. What happens if you try to stand still on a bike for any length of time? You kind of just fall over, right? So you become destabilized. And with this RNA polymerase, that's exactly what happens. It starts dissociating from the DNA template itself, and then it lets go of that mRNA transcript as well. So it loses its binding to both of those, and it kind of just falls off at that point. That's how termination happens, and that's because it's reading a signal as a stop signal on an mRNA transcript. Okay. You have mRNA, you have a transcript, and it's complementary to which strand, template or coding? Complementary. Template, yes, it's complementary to the template strand. And it would be exact, more or less, to
Minus the uracil.
minus the uracils and thymine leads, yep, okay, exact to the coding strand. All right, now we've got our transcripts. We're going to be talking about translation, what happens now. With prokaryotes, this is a coupled process that happens together. So transcription and translation occur together because you don't have a nucleus, you don't have a membrane separating this space. So as soon as this gene is transcribed into an mRNA transcript, that would be like this fuchsia line that's coming off your mRNA transcript. As soon as you get that mRNA transcript, boom, you're hitting ribosomes that are right there in the cytoplasm, and those ribosomes are going to town, starting to produce those polypeptides from those transcripts. And eukaryotes, again, this is totally different. It's much more complicated because you do have that nucleus. When you access those genes and start transcribing in a eukaryote off to the right there, those transcripts have to have a bunch of further work done before they're ready to be translated. They have to have a 5' cap, they have to be 3' polyadenylated. There's usually splicing out of intronic sequences, and then they have to get exported by chaperones out of the nucleus and carried over to the ribosomes. None of that has to happen with prokaryotes, because again, like chromosome is kind of just out there in the cytoplasm. So as soon as that transcript comes out, it meets ribosomes and it starts translating. Coupled processes. Yes, sir.
So do bacteria not have intron and exons?
They do not. That's a feature of eukaryotes.
Piggyback. Go ahead.
Yeah. Oh no, piggyback, and then I'll add on to something I was going to say there.
So do they not have introns and extrons, or they just don't have the ability to take out the junk? I guess,
They
like,
do not have introns.
Okay.
So let's say you have a prokaryotic bacterial gene that is 999 base pairs long. The mRNA transcript that gets processed and translated by the ribosome is going to be 999 base pairs, but then the protein itself is going to be how big?
333 amino acids.
It depends on what. Okay, but approximately?
300.
It's
gonna
going
be
to be divided by 3, right? Because you have a
333.
triplicate code. 3 nucleotides for every amino acid. So about how many amino acids big is that protein going to be?
333.
About 333. With a eukaryote, with all the introns that are in that message that need to be spliced out, which we're not even talking about in this course because we only care about bacteria, the math doesn't work. It's not divisible by 3 until you get to the part where the mRNA has been spliced. Then you can divide it by 3 and you can calculate approximately how big that protein will be. But with and that would be a fine exam question too. If you have which of these answers would be most correct for the size of a protein coming off a prokaryotic gene. Okay, but does that make sense to everyone? Yes.
Just to be clear, now, you were saying, so for prokaryotic, they will be in replications of 3 because they don't have any of the introns to splice up?
The codon, yeah, so that codon code with prokaryotes is much simpler because there are no introns to splice out. So it's preserved, and again, like the size of your gene that's transcribed on that transcript is going to be reduced by a power of 3 when it gets translated into amino acids or a polypeptide. Yeah. Okay, so this figure kind of hopefully shows you the main differences in these processes, but takeaways: uncoupled processes, coupled processes, talking about transcription and translation. Okay. All right, so when we get down to translation, brass tacks, now we're back in the ribosome. which we said was about 60% ribosomal RNA and about 40% protein. We have these three different sites in the big ribosomal subunit, the 50S, that correspond to different actions and activities that have to happen in order to get this growing chain of polypeptides. The 30S ribosomal unit has a different function. So if you kind of think about this like you would think about your hands, 30S would be the hand that's holding the mRNA. So here's your mRNA, here's your 30S subunit. It's going to seat right there, and it's going to read the mRNA right there. The 50S large subunit comes and it covers on top of that mRNA, and that 50S subunit is responsible for actually making this growing peptide chain. So between these different amino acids that are coming in, they all have to be strung together to create peptide bonds between them. And that transpeptidation is what's happening and occurring in that 50S subunit. Going back to those sites here, so we have our mRNA that's being read. The A site, aminoacetyl site, is being read. UUC is going to be complementary to that tRNA. The tRNA is what is shown right here. So this is an incoming brand new tRNA that corresponds to that codon. So there's the anticodon, and then there's the corresponding amino acid, the phenylalanine. When it comes and sits in, if it's a good, appropriate anticodon-codon pairing, it'll ratchet over to the P site, the P-peptidyl site, and this is where that phenylalanine will be joined to the tryptophan, the preceding amino acid in that chain. After that's happened, it is uncharged, so it's released its phenylalanine, this particular one, and it gets kicked out, rejected from the ribosome. That uncharged tRNA can go pick up a new phenylalanine at that point and then re-enter the process. But that's what we call tRNAs that are no longer carrying an amino acid. We call them uncharged. They pick it up again and they're charged. Okay. So they're just kind of reading that messenger RNA until they hit which codon? The stop codon. Okay. And that's when this entire thing just kind of stalls out, loses momentum, destabilizes, dissociates. Okay. This slide on termination. So there's a little GIF that kind of shows you the movement of tRNAs and how they're being joined at the 50S ribosome. But mRNA continues to be fed through that ribosome 5' to 3' until they reach that stop codon. And then usually that whole ribosome will just fall off and dissociate from that messenger RNA. Average protein size is usually somewhere in the order of between 200 to 300 amino acids. Some are quite a bit bigger, some of them are smaller, but most of them are in that size range. Okay. And I think we'll end today talking about gene expression. There's more in this deck, but we'll pick up with that stuff tomorrow. This is hopefully pretty intuitive, but when we talk about bacterial gene expression, there are some genes that kind of need to be expressed all the time, and there are other genes that don't need to be expressed all the time. The cell receives cues from the environment that tells it if it is lacking something or if it has too much of something, and then it can respond appropriately. But when we're talking about so-called housekeeping genes, things that are involved in energy production or trafficking of proteins around the cell, really, really important functions that never really stop being necessary, those are going to be constitutively expressed. Constitutive expression means you're pretty much always going, you're always turned on. You might be able to bump up levels a little bit, but it's never really turned off. So that's what a constitutive gene is, housekeeping genes. There are two other categories of genes, inducible genes and repressible genes. So with inducible genes, these things are going to be off until they're turned on or induced. What does it mean to induce something?
To activate it, like induce labor, you induce.
To cause it to happen, to provoke, to effectuate, to potentiate. So if you induce something, it's not doing something and then you make it do something. It's an inducible gene. The other category are repressible genes. These things are on. They are turned on by default. It's their default setting to be on transcribing. You have to turn them off or you have to repress them. otherwise they stay on. There's some classic examples that we'll go through that'll underscore this, but just a couple terms first. So here we have a schematic of some genes, some bacterial genes. And this is kind of unique to the way bacteria express their genes. Eukaryotic cells don't do this. But what prokaryotes will do is put a bunch of related genes together that have a similar function and they need each other in order to function properly. They'll put themselves together in a cassette called an operon. So an operon is exactly what is up here. These are related genes that are just transcribed as a group together. We also have upstream of that operon a regulatory gene, which can get expressed, and this usually will encode for activators or repressors that will have either an inducing or repressing effect on that gene that's downstream. And then you have the promoter site, which is also upstream of the genes. This is where the RNA polymerase will bind when it can transcribe. And then an operator site. This is where repressors will sit. When those repressors are transcribed from these regulatory genes upstream of those, this is where they sit, right on that operator. And what they do is they basically provide steric hindrance for that RNA polymerase. We've talked about steric hindrance before. We were talking about it with antibodies, but what does it mean if you have steric hindrance? Something? Yeah, it's like a big physical blockade. So if you have a repressor sitting right there on an operator, what you're physically blocking is the RNA polymerase from doing its thing and transcribing those structural genes. Okay. So that's how that works. And then when the operon gets expressed, all these individual structural genes, they will come off on a single mRNA. This is a polycistronic gene. And then when they're translated, those can be cleaved by proteases into single functional subunits. Okay, so a single transcript that cleaved through proteases into like protein A, B, C, or D. See an example. It'll make more sense in a second, hopefully. Okay, so this first example is talking about inducible operons, which, as we said, are operons or the genes in operons that are going to be off until you induce them, inducible genes. So they're under positive control and they need some sort of activator in order to have transcription. The classic example is lac operon. Many of you have probably heard of this example before. You've taken cell biology classes or like microbiology classes before. But in bacteria, bacteria are starved of glucose, which is their preferred carbon energy source. They can use lactose. The problem with lactose is it is a disaccharide, so you need to have some carbohydrate enzymes break down that disaccharide into glucose into lactose. But if you don't need to do that, then you're not going to waste the energy, expend the cost at expressing these genes. Because look at this, in this operon you have 3 genes. If you don't have lactose present, why expend the energy? This is an energetic process, and that's why you put this under inducible control, because it's not something you need unless you need it. Okay? So in this example, these genes are expressed in the presence of lactose, which means the cell can use it if it can break down that disaccharide and use that glucose. These are 3 structural genes that are involved in the breakdown of lactose into glucose and lactose. And what happens is that by default, as an inducible gene, this thing is off. So this cartoon is showing that there is a repressor, a LAC inhibitor, that is binding to that operator site. Can you see that? It's binding to that operator site. And it's blocking the polymerase from transcribing the genes in that operon. That's what that red block means. Okay. In order to get it to actually transcribe those genes, you would have to induce it. And you would have to induce it by the presence of lactose. So when there's lactose present in the system, that lactose will convert into this form called allolactose, which will then bind to that repressor protein. When it binds to the repressor protein, that repressor can no longer go do its job. It can't sit on the operator anymore, which means the RNA polymerase is then free to transcribe that operon. And then voila, you get all 3 genes in that operon expressed, and you get the ability to now break down the lactose that's present in that system. very briefly, just to explain how repressible operons work, they too are in response to environmental conditions. In this case, though, they are on unless they are turned off or repressed. So in this case, the tryptophan operon is the classic example. And this is sensing levels of tryptophan in the cell that bacteria need. If they don't have tryptophan in the cell, then this repressor that you see down there is not stable enough to bind to that operator site. And because of that, you get continued expression of the genes in that operon, the tryp genes in that operon. When you do reach a critical threshold of tryptophan in that cell, it's time to turn on expression. It's time to block expression. You no longer need those genes. So the tryptophan will bind to the repressor, and it will become stable and able to bind to that operator. And once it binds to the operator, it's the same phenomenon as before. You have steric hindrance, and you no longer have expression of those genes of that operon. It's repressed. That is it for today. That's all I was wanting to cover. We'll pick up on mutations tomorrow. But before I call it a day, do you guys have any questions over that material? Okay. Yes, sir.
Going back to the inducible operons, just on the second slide after that, the next one.
Okay. This one?
Yeah, which is the which one is the activator protein on this?
There's no activator on this schematic. This is just showing you what what's happening to the repressor.
Okay.
Yeah. Yep. Does that,
Operons are just like tools that the bacteria has to be efficient.
yeah,
So it
kind of.
doesn't waste
So,
energy.
so for the example, like these three proteins coming off the operon, say they weren't expressed together as an operon, say they were expressed separate individually, you would never find a purpose for this beta-galactosidase. And same with permease, they work together. So you express them together. There are other examples of pumps that need to work together, that have separate constituents that work together. They get expressed as an operon. So think of an operon as something that needs to be expressed in order to have complete functionality. Yeah. Related genes, related in function and purpose.
Key that turns on the mechanism.
Yeah.
Kinda?
Yeah.
Like a grouper.
Yeah.
So, endospores, like the ability for certain bacteria to sporulate, is that an inducible gene or is that a repressive gene?
So say you have a spore-forming bacteria, like B. anthracis, and it receives some environmental stimulus that tells it to sporulate, to turn on these genes. It might be more complicated than even a single operon. There might be multiple operons involved. So it is, sporulation is kind of a big deal. You have to, it's not just structural, there's also a lot of regulatory things that are happening during sporulation. So, there's another question over here?
I'll ask you after.
Are you sure? Okay. All right, well, thanks guys for your attention. I know it's been a long day already, but do come up to me and let me know if you have any questions.